Thank you for the introduction. I work at the Silesian University in Gliwice, Poland, and the other contributors of the work I'm presenting here are Andrzej Zieleziński from Adam Mickiewicz University in Poland, Cedric Notre-Dame from Center for Genomic Regulation in Barcelona, and Sebastian Deorowicz, chief of our group from our university. So basically, the motivation is probably well known. Due to large-scale sequencing efforts, protein databases are about to explode to billions of entries. And the tools that supports discovery of protein families, so basically the similarity, sequence similarity or structure similarity search tools like Diamond, MMSex, or TFOLDSeq, they are able to handle that amount of data, while multiple sequence alignments do not. So basically, which are crucial for phylogenetic reconstruction or function prediction, domain identification, and many other applications. So most accurate methods scale poorly, above 1,000 sequences. There are fast algorithms, but they are not that good in terms of accuracy. There's a lot of noise in databases, which hits accuracy as well. And actually, no method is ready to handle families with tens of millions or more proteins. Well, I've just realized that I should have put here a picture of multiple sequence alignment, but fortunately it was at the previous presentation, so MSA Transformer saved me. Thank you. Okay, so to address these issues, we present FAMSA 2, which is a successor of our previously published algorithm. It's a progressive algorithm, meaning that it consists of two stages. The first is construction of so-called guide tree. So we can think of a guide tree as an approximate phylogenetic tree, but we on purpose do not call it like this because it's not like phylogenetically correct tree does not necessarily lead to the best sequence alignment, yes, because these are all heuristic algorithms. And then the second step is to align sequences in the order described by the tree. So here we have a flow chart, of several use cases, but the most important, the most typical is the one shown in black arrows. So we go from unaligned sequences up to sequence alignment, yes, with calculation of intermediate guide tree. This calculation involves also a computation of dissimilarities, which is kind of important from the point of view of accuracy. So regarding this dissimilarity, in our tool, it is based on the longest common subsequence, LCS, not to confuse it with longest common substring, because longest common substring is when we have the letters one after another. In subsequence, it can be spread for the entire length of sequences with some unmatched residues in the middle. So actually, we can consider this as a global alignment, sequence alignment with unit match score. Yes, basically. Actually, the measure also considers unmatched residues, so there is a penalty for residues which do not constitute to the LCS. But what is important is that since we have a unitary match score, it can be encoded with bits. Yes, basically, it's a binary information. And this alignment, there are dynamic programming algorithms which operates on bits, which are very fast. And if we combine it with vectorization on modern CPUs and multi-threading, it turns out that we are able to calculate compare tens of thousands of residues at the same time, which translates to hundreds of millions dissimilarities per second on modern hardware, which is kind of quite a throughput. Another thing is that the measure turned out to be robust to sequence divergence. I'm going to talk about later. Okay, do not look at this. This is just for illustration. Yes, it's way too complicated to talk about it. Okay, and regarding the guide tree, FAMSA uses single linkage tree, meaning that at each step, it just combines the two closest sequences or subtrees. And in order to generate this tree, you have actually two options. The first one is to calculate the exact tree using Prim's algorithm from minimum spanning tree, because minimum spanning tree can be easily transformed to single linkage, which is quadratic time, not very appealing in terms of scalability, but unlike UPGMA or other type of algorithms used by sequence alignment algorithms, it's linear time, and meaning that it does not need the entire distance matrix at a time, only a single row, which is actually important from the point of view of scalability, yes, because, well, quadratic time, you can always wait longer, yes, but if you have quadratic memory and the matrix doesn't fit into your RAM, there's no way, yes, to go. You have no other options. Alternative, and that's, let's say, one of the major contributions of this package is approximated tree, which we called Medoid tree algorithm, which, as you can see, scales as n log n with a number of sequences. Yes, the large n is the number of sequences, times m, which is an algorithm parameter, basically. Yes, but you will see that actually it scales much better. It's also linear memory. Both these algorithms are parallel. Okay, so how these Medoid tree algorithms work? It's a very simple divisive clustering procedure, which was inspired by mapped part tree. Yes, in the sequence alignment package mapped, they introduced like 15 years, maybe more years ago, a very scalable solution for guide trees. The idea is that we wanted, because mapped is a very random procedure, this part tree algorithm, we want to somehow reduce this randomness, this stochasticity, by replacing random so-called seed sequences with medoids. So the idea is very simple. We have a set of sequences. So these are these gray points. We select randomly some sample, perform partition around medoid clustering to establish seeds. Then we build a parent tree on these seeds. Well, it doesn't look as a tree here, but actually it's a single linkage tree for these 4 sequences. And then we assign other sequences to the seeds. So we split, yes, data set for parts. We generate recursively subtrees, combine with parent tree, and so on. So it goes like this. So actually, the major difference compared to mapped part tree is that it selects randomly these red points. Yes, meaning that this procedure, the topology is very unstable, meaning if we reorder the sequences, we would get very different guide tree, yes? And what we wanted to do, we wanted to stabilize this procedure a bit, reduce the stochasticity by introducing this medoid clustering before, yes? So it's not like it's entirely deterministic, because we still draw some sample, but it's more deterministic, let's say. There's less randomness, yes? And actually, when you look at the seed representatives of the seed sequences, I have a family here. Yes, it's a PFAM family. I intentionally selected the picture for which there were the best results, obviously. So you can see that there are some clusters here, yes, within this family. And if we draw the seeds randomly, some clusters are not represented. Yes, some are overrepresented. But if we perform this medoid clustering before, so draw some sample, do some medoid clustering, they are better. They are plotted using multidimensional scaling, yes? And actually, this translates to another important feature, which is topology stability. Without going into the details, here we have Robinson fold distances, meaning lower values, the larger stability. The procedure was that we took some sequences, core sequences, added some another, made an alignment, generated the guide tree, then pruned out non-core sequences, and we checked how they are similar, yes, between replicates. And clearly, when we apply this medoid step, the stability is better, yes, compared to part tree. We have our implementation in order to have consistent dissimilarity measure, and we also use the original mapped variant. Okay, so in order to evaluate the method, we performed a set of experiments. The first involved reference-based evaluation, structural reference-based. So we generated an updated version of EXT-HOMFAM, extended HOMFAM benchmark, which actually was at first introduced in Cluster Omega study. They call them HOMFAM, and it consists basically of PFAM families, in our case with up to 3 million sequences, the largest one, with Homestrat reference alignments. And thanks to the presence of reference alignments, we are able to calculate the fractions of correctly aligned residue pairs, which is sum of pairs score, and total columns, yes, basically. So we have the reference. And when you look at the methods, yes, so there are a few algorithms, alternative solutions. You can see that you can like divide them into two groups, like more accurate. So these are like bottom 5, starting from muscle 5, and less accurate. If you look at ECCB, SP, and TC scores, there's like 15-point differences in this percentage point differences. The thing is that when you look at the execution times, all methods beside K-align, they struggle. So they failed at the majority of them failed at the largest family. And yeah, they need like a week or 2 weeks or even a month to finish the entire benchmark, while our software needed 15 hours, rendering the best results. But what is most important, if we turn on this approximated tree feature, we got very decent results, like similar to Muscle 5 in 47 minutes, yes, for the entire benchmark. So it was even faster than K-align with decent accuracy, yes? Okay, this is just to show when we stratify this benchmark into subsets with increasing size of families, you can see that this distinction between less accurate and most accurate method, it become more pronounced as the data size increase. And also this approximated tree version, medoid version, has the largest scalability in terms of execution times. Yeah, so it just rise the dynamics of the increase is the smallest here. Okay, another test. This was kind of, I think, interesting experiment. What we did here is that we took largest families, yes, above 1,100 sequences. We subsampled them at some points, yes, 100, 200, up to 1,000, and then we mixed all the families. So it's like very unrealistic test case, but it shows some properties of the algorithm. And then we aligned, yes, so we have a back full of families, and then we run a multiple alignment procedure on this, and we evaluate the families independently, yes. So actually for every value, we have only a single data set, yes, which scales up to 12 million sequences, yes, because it's 100 families, yes, with 100,000 sequences. So besides scalability issues, you can see that there are missing points here. Some software failed to process larger data sets. So actually FAMSA was the only one to process all of them. You can see that that was the only software where the accuracy increased. And when you think about it a bit more, it's kind of intuitive, because it's like we have these families are some clusters, yes? And if we have only reference sequences, by reference, I mean only this Homestrat sequences with known structures. So we have like 5, 10 maybe sequences for each family. So we have these, let's say, structure, and we sample this data very sparsely, yes? And once we increase the number of sequences with each family, so these clusters become more visible, more pronounced, and alignment algorithm is able to somehow like use them to cluster this properly, yes? And it turned out that this is due to the similarity measure. In FAMSA 2, we like decreased a bit the penalty for unmatched residues. So that was the modification in this step, actually. And it turned out to be crucial for this behavior. So here what we have is that we selected sequence pairs from this mixed data set. And if we performed random selection, and we measured how far they are in the guide tree, basically. And if we performed random selection, you can see that there is no difference between FAMSA 1 or FAMSA 2 at all. But if we take the pairs of sequences which are related somehow, you can see that adding homologs like reduce this distance in a tree. Yeah, so these additional sequences helped somehow in properly clustering the sequences from the same family. Okay, the largest disadvantage Okay, I'm running out of time, I can see. So I'll move on maybe to phylogenetical evaluation, because multiple sequence alignment tools are very often used in phylogeny, which is hard to, it's hard to like evaluate phylogeny on large scale due to lack of reference phylogenies, yes? So what we do is we performed some indirect evaluation where we just compared the topologies of maximum evolution and maximum likelihood trees. So the rationale behind was that if these trees agree, it means there is less noise in the alignments. Yes, basically. And you can see that here we have this tree agreement plotted against structural accuracy. Finally, function evaluation. This was just to measure how active sites are spread across alignment columns. So ideally, they should be like in a single column. But well, evolution and function are different things, so it's not always like this. Okay, just to conclude, in general, what we present here is the method that generates superior accuracy in the tests we performed, combined with unmatched scalability. Namely, the largest family was processed in 5 minutes, yes, with this Meadows tree variant. And well, it may be a bit overstatement, but we can think that this method is kind of ready for future challenges in comparative genomics. Okay, so as for availability, you have a preprint GitHub repository and very old publication with the first version of FAMSA. So thank you so much for your attention, and I'd be happy to hear any comments.
Okay, I think we have time for one question. So this was a wonderful talk. Anybody? Oh.
Yeah, very nice talk, and it's really great to see work being done on improving alignments at this time. Do you think the benchmark, the Homestrat benchmark, which was built at that time? I mean, I checked. I mean, it's a long time ago, based on the handful of structure that were available at that time. Do you think maybe a bit superseded? Is there an opportunity, you think, to revisit this?
Yeah, I agree with you that it's updated. They are like doing releases, I believe, every 3 months, but they do not change at all since, like, I don't know, 20 years ago. So I agree. Yep, that the thing is that we do not have much more. Yes, so that's why we perform also different types of evaluation. Yes, so
we have more structures today, no?
Yeah, I know that. Yeah, but the thing is that we need to, we would need to like to take the structures, align these, yes, using structural alignment methods, and yes, I agree.
Unfortunately, I think we're going to have to move on to the next talk, but